Thank you for inviting me for this CME, and I hope a lot of the members and the office bearer of the Society of Renal Nutrition and Metabolism Associations. Without wasting my time, let me introduce. I just start with the quote by Professor McCanns and Widdowson. It's a knowledge of chemical compositions of any food is the first essential in dietary treatment of any disease or in any quantitative study of human nutrition. So, understanding the food component or the nutritional component of food is very important wherever we have used for various applications, including treatment of various diseases in human nutrition. And if you look at the food composition, the food, whatever the food which relevance to in case of a development for various other applications, and study on the foods which covering the methods, procedure to determine the properties of food and its constituents, all deals with the food analysis. Then when the properties of the foods comes, there are so many properties which includes the food composition, the structure of foods, physicochemical properties of food, and the sensory characterization of the foods. All those things it deals with the properties of foods. But today and my area for now work exclusively on the food composition, so that I'll be taking more deep on the food composition, particularly how we developed, because that is very important to discuss more on the development part, because you will understand how we have systematically planned. Coming to the other, the food composition analysis, the description, it's nothing but it's a chemical analysis which included maybe the macronutrients, micronutrients, other phytonutrients. Now a lot of importance are being given for the phytonutritional database development as well, like polyphenols, carotenoids, vitamin C, etc. And also have given emphasize on the anti-nutritional factors like phytate, oxalate, trypsin inhibitor, so on. And the food composition tables are the database, also called it's a repository for the content of nutritionally relevant chemical constituents and energy value of any foods. And this food composition table also relied upon the nutritional research and the health practice, as they provide critical information regarding the content and distribution of various components in different foods. So the construction, development and management of the FCT involves statistical designing, the food sampling, component characterization, and the selection of methods and how we are using methods, appropriate methods, and the estimation of various nutrients, and the data quality management. What kind of quality management we are incorporating in an analysis, and the documentation presentations, or these are the important components, if you wanted to construct any national level database on the food composition. There are different types of food composition available across the world. The first one is first and very important food component food composition table is the original analytical value. It's nothing but it's a published literature or unpublished literature reports. It's directly coming from the laboratory, whether or not they were from the analysis carried out at, the explicitly for the purpose of compiling or not, but we can use as a data. We can compile all these published data which is originally analytical data. Most of the data have been including USDA, even the Denmark database, and Canada database. There are so many databases. It has been compiled by these kind of the published data. Then another type is imputed value, where it can be derived from analytical value obtained for similar kind of foods. So for example, if you wanted to get the dry peas or the fresh peas, and it can be taken from either fresh or dry respectively. Or even you wanted to take it for the defatted, we can derive from fat, with fat samples that can be imputed like that. Then also can be calculated values. There are some data which can be derived directly from the recipes, we can back calculate and we can impute in the database. And borrowed values, yes, of course, if we don't have the data, we can borrow from neighbor country. For example, Bangladesh people, they have borrowed our data, which is considered, it's a close to the west part of our data. So we have shared our western part of our data. Similarly, the Sri Lankan uh, people, they have developed recently, they have published Sri Lankan food composition tables this month, the first week of last week of November. And we also have shared some of the data which they don't have, but the food which is commonly consumed both in the southern part of India and Sri Lanka. Similarly, like this, no, we can borrow the data also. The presumed value, sometimes we can presume the value, or otherwise we can consider as a zero, and it is very rarely will happen. The application or the user of the food composition data, if you look at, there are variety of people they use it. That we have to understand, not only the dietitians or the nutritionist what we practice, but apart from that, there are so many other applications essentially using our food composition tables. The first one is the assessment of nutritional intake at the national nutritional analysis level. And now, this is the value generally we used to take it for any kind of nutritional intake, dietary intake data, to calculate the dietary adequacy or dietary inadequacy and dietary imbalance, dietary advice, and the prescription of therapeutic diet if anything is required. And that is for the individual level. And coming to the group level, in case of any different physiological groups, growing children, NPNL, women group, socioeconomic group, clinical group, like this various groups also, the nutritional intervention programs, or if you want to calculate anything regarding the dietary adequacy or intake, these kind of database will be essentially used. Then at the national and international level also, the individual nations can be assembled to give the cross national or worldwide picture of the food and nutrient availability, to formulate the food and nutrition policy, setting up a goal for agriculture production. So based on the availability of the nutrients also, a different country they set up the agriculture production and the guidelines for the consumption, policies for food fortification and food supplementation also. Even we are closely working with the FSSI and to formulate so many fortification programs and the supplementation programs. All it's based on the food composition of commonly consumed foods. And internationally, yes, it has implications for trade and for the development assistance policies, and this kind of database has been internationally used. And planning, advising, and prescribing food and nutrient intake at if you look at the physiological requirements, the recommended intake of most of the nutrients, recommended dietary intake of most of the nutrients, even internationally, like FAO, WHO, even also used to formulate that can be translated into varying food intake at different levels. Like if you wanted to prepare a therapeutic diet, so especially the therapeutic diet, we need a lot of practice, and practice and detailed training on this application of or understanding on the food composition. For example, if you wanted to prepare for the diabetic mellitus or diabetic based diet, and then we need to understand the energy, glycemic carbohydrate, the fat, protein, and dietary component of the foods consumed needs to be understood very thoroughly. Similarly, anemia means we should understand the iron content, iron composition of all the foods, and likewise, even for the our renal problem or the nephrological problem, also the phosphorus, Dr. Anita mentioned, phosphorus and the potassium. Understanding the phosphorus and potassium is very very important. We have to understand if you wanted to.
Hello, if there's a lot of noise coming, would you like to mute yourself? Excuse me, who are you? Yeah.
Yeah, I think they have muted. And similarly, planning of institutional diets. Suppose if you understand the component, food composition of different foods, you can able to make it's a cheap or the limited source or with the low cost cost based foods for various sectors like military, cafeteria, hospitals. Even NIN is formulating for our state government and various other government institutions with the lowest price without compromising the nutrient which is required for various group of populations or the very age group. And the national food and nutrition for policy, and we have to define the goal for intake of certain nutrient that must be translated for food productions targets, as we saw in the previous slides for agriculture sector, or into food consumption target for market and public health sector. Because if you wanted to give any increase the subsidy or promotion of certain foods, and without compromising, and you have to target certain amount of foods, and based on the productions this can be planned, the increasing, if you want to do increasing or reducing the subsidy or promotion of various foods. Similarly, the nutrition regulation of food supply, the food regulations uses nutritional or food composition data of primary or traditional food products as a reference point for desirable nutrient level for the processed and the newly introduced foods. So like this, even for the government employees, is the fixing various the DNS allowances, the government policies, many policies also they use the food component and essentially which is available in the commonly consumed foods. And also it's used in the check on the label information or claims most of the packaged foods they use it. And the planning of nutritional intervention programs also is one of the important utilization or application of the food composition, where the it translates specific needs into food requirements, and food aid programs or supplementation programs used with the food composition data. So there like this there are so many applications are there. I don't want to go very detailed one by one, as you know very well that. And I just wanted to give the the wide range of applications, because everybody we use the food composition table. So in order to make uniform database, the worldwide FAO recommends us a lot of the procedure. If you wanted to develop a national level food composition database or tables, so we have to follow that, so that systematically we have followed for the development of these Indian food composition tables. I'll take you through the development of how we planned and how we executed the development of food composition tables. As Dr. Anita Madam says in her introduction remark, it's not a new one. This Indian food composition tables are the nutritive value of Indian foods. We started the NIN used to be a it's the main center to develop the food composition tables. It started way back in 1937 by Akiod, and our institute were in Kunur and Udagai, Uti, and that time it was nutritive value of Indian foods and the planning of satisfactory diet. And then later on, a lot of revisions from 1937, 60, 63, 66, like that. So many revisions have been taken place, and then the last revision was done in 1989 as a nutritive value of Indian foods. And with the passage of time, and due to improvement of a lot of analytical techniques and change in the food supply and variety of foods, and the proportion of the data that are considered to be reliable decreases. And again, keeping in the mind that changing scope of range of nutrient and bioactive substances in the foods, and their importance in the health and diseases, and generating a new database or the device revising the food composition tables, it was necessary to cater the need of users and following the systematic guidelines. um, Yeah, this is the first what we developed. It's a health bulletin as a nutritive value of Indian foods and the planning satisfactory diet in 1937, with 208 foods with and containing 14 nutrients, and subsequently 1951 30 nutrients and 229, and another the third revision in 2000, sorry, 1971, with the 664 foods with the 440 nutrients, and it was the last revision by Gopalan and et al. In 1989 with the 597 foods containing 57 nutrients. Then later on, the whatever we have revised and released in the year of 2017, after many years, it's as an Indian food composition tables, which we are going to see in detail. So in this, the food composition table development, it involves so many stages, collection of the samples and the identification of the samples, and then how we prepare the samples, analysis, maintaining the quality controls, both external quality controls and in-house quality controls, and analyzing the data, then the quality, data quality review or data cleaning, and finally is publishing. It's only the bird eye view of how the different stages involved in the development of food composition. The first and foremost stage for the development of the national level food composition table is identification of key foods, because our Indian cuisine varies widely across the country according to the region, culture, and tradition, and characterized by the use of different spices, vegetables, grains, fruits, vegetables, and variety of foods also, and then the animal foods also, animal sourced foods. So therefore we cannot collect all the foods, because it consumed in the whatever the consumed in the country, we cannot collect it, because the feasibility and the cost involved for the analysis also needs to be considered. Therefore, the best approach to collect the food is the key foods approach, already developed by the USDA, by the Hitovich et al. And they also followed for their development of USDA food composition table. So the same approach, we also adopted the key foods approach. The key food is nothing but the food which contributes up to 75% of the nutrient intake by for the population. So what we did, the method combine this, actually this key food approach combines composition data along with its nutritional nutrient composition, and then the ranking the foods by applying the scoring system, then followed by we can identify the foods which will contribute significantly to the diet in terms of the nutrients. So based on this systematic arrangements, we have identified the foods already we have published in our book as a key foods. Then for that, for our identification of our key foods, we have used our National Nutrition Monitoring Bureau (NNMB) database 2006. It's a rural database, and where so as you know that NNMB, it was functioning under our NIN, one of the branch at NIN office, but right now, though it is not, it's working, but the data what they have collected in terms of the dietary intake, it is a huge, which was covered almost three-fourth of our geographical Indian region, or two-third of our population, and both rural and urban data. It is a huge data. Even we are using it for many other correlations and applications of intake or the nutrition status of various population, even right now. So we have used the latest 2006 data, which was conducted in more than 30,000 individuals in all the age groups, using 24 hours dietary recall method, and that was the data which we used to select or identify the key foods. Then after identifying the key foods, the next stage is how to collect a sample from different parts, because if you look at our Indian regions, a lot of climatical zones are there, agroclimatical zones are there, soil types are there, So a lot of ecological variations are there. Again, the food, the variety of foods or the species of the foods, different cultivars of the foods also varies across the country. So now, from where we have to collect the samples, so we have to follow adopted method or internationally accepted method. So we use a national representative sample planning, and it was based on the probability proportional to the size. So the method we used, based on the probability proportional to the size, national level sampling plan, we have developed based on the FAO already what they have recommended. And because we cannot go based on the population per se, then because the population, it was varying depending upon the state. So then again, without changing the probability proportional, then we have restricted to the number of district per state. So based on the number of based on number of district in each state, so we have statistically we have selected. I'll show you how we have identified based on the number of states. So essentially, based on the probability proportional to the size at the district level, we have identified the sample collection center. And again, we have divided our country into 6 parts. And these same kind of the divisions or the segregation of the parts, 6 parts, also have been adopted by NFHS surveys, except the Uttar Pradesh, and we have used in the central region, whereas it was in the north region by NFHS. So these are all the different the part or the regions what we have um, divided, which in the central part, Chhattisgarh, Madhya Pradesh, Uttar Pradesh, and Uttaranchal, whereas in the eastern part, Bihar, Jharkhand, Orissa, and West Bengal. Northern part covers Chandigarh, Delhi, Haryana, HP, Jammu and Kashmir, Punjab, and Rajasthan. Whereas in the northeastern states, all the states, seven sisters, Arunachal, Assam, Manipur, Mizoram, Meghalaya, Nagaland, Sikkim, and Tripura. And then south, we cover even the Andaman Nicobar, Andhra Pradesh. It was United Andhra Pradesh that time. Karnataka, Kerala, Lakshadweep, Pondicherry, and the Telangana and Tamil Nadu. And the west, the Daman and Diu Daman, Goa, Gujarat, and Maharashtra. So these are all different states, and which covers under different regional part. And this is what actually we have selected a number of districts based on the availability of the district in each states. For example, if any state having 1 to 5 districts, and we have to select only one district. So such a way that if you look at 1 to 5 states, there are 9 states where having one to between 1 and 5 districts. Therefore, in each districts we have selected one. It covers 9. So like that, there are different states which falls under different districts. Based on that, we have identified number of districts needs to be sampled. It's like almost it's covering 107 sample collection spots or districts. So these are all the different sample collection centers identified at northern zone. Then coming to the southern part, it's a southern part, whatever the red colored dots, which gives the sample collection spot or sampling center. This is for west, and this is for east. Sorry, this is for the west, and this is for the east, and this is center and the northeast. So like this, the 6 parts: north, south, east, west, center, and northeast. After identifying the sample collection sampling center, then what we did, these are all number of the states, and identified selected districts for the sampling, including union territories. These are all different union territories. So For the north, it's a 23 centers, east 20 centers, and south 19, west 12, and central 22, and northeast 11, which in each regional center, which covering 107. So after identifying these centers, what we did actually, at a time, uh, our people, more than 107 people, in our project itself, almost 90 people we worked, including starting from attendant to senior technical officers and scientists, and our office were permanent employees all together. We have prepared the list of foods which needs to be collected in each batch, with identifications, proper identifications. And if they have any clarification or the confusion, then we also have taken its as sample, little amount of sample in a pouch. So we made a list of samples to be collected, along with the all the materials, packing materials, labeling materials, all the stuff, along with all the stuff. So all the people from Hyderabad uniformly dispersed to all the respective collection center in at a time, because in order to avoid the seasonal variation among the foods. Then after reaching the place where the samples needs to be collected, supposed to be reached in at least one day in advance, in order to verify the sampling or the markets where the more people are purchasing, at least at different three different levels. So one is in the supermarket, and one is in the middle, and one is in the retail price. So like that, the people will identify one day in advance, and then after identifying the sample collection or the stores, the purchasing center, then the people will collect the sample in the same day in all the places. And then after collecting the sample, the respective to each region, they will come to the identified zone. In the sense, the each zones, again we have identified one particular place for compositing the samples. Let's say for north Delhi, and for center Varanasi, and east Kolkata, west Mumbai, and south Bangalore, and for northeast it was a Guwahati. So they have transported along with the sample. They reached at a time to the respective sample compositing center in each region. So then after compositing there, suppose when we purchase dal in each collection center, it is 2 kilo of dal. Then that needs to be, we have screened whether it is the same variety or the same cultivar, and if it is any difference is there in terms of the morphological identification, and that was analyzed independently. Let's say the brinjal, there were 23 varieties of brinjal. So otherwise, if you are able to make composite, if it is a similar morphological identification, so that we have pooled it together, and then from the pooled sample, the composite samples, only approximately, if it is the vegetables, 2 to 3 kg, or the dry grains, it was again so 1 to 2 kg of dry foods, it was transported to Hyderabad with the proper packing techniques, including the green leafy vegetables or the vegetables without spoiling or the freshness. So we have adopted very good packaging techniques. Based on that, so we have packed all the foods. It was transported to Hyderabad for analysis. This is another one representation, how we have selected the centers. If you look at, there are 6 regions, as I said, and coming to the Madhya Pradesh, there were 50 states. So in between 50 to 60, and it's supposed to be 8 different districts. And similarly, in whatever in the center, the Madhya Pradesh, Uttar Pradesh, Uttaranchal, and Chhattisgarh, from all the 4 states. So, and in each states, we have number of districts we have given. All together, it was pooled at a composite sample, where the center belongs to the Varanasi. From that, we have taken the sample, and finally, it was analyzed in a duplicate. This we have divided into 2 parts, and one is original and another one is duplicate, in order to understand the consistency of the nutrient analysis. Similarly, like we have collected so many foods, I think you might be seen in our food composition tables. We also have given the photographs. Even we have purchased different poultry products, And even in the poultry products, different parts of poultry, chicken meat, and the meat and meat product are the inland fish and shellfish. What we did, we have not collected from all the 107 sample collecting center or the sampling center. Instead of that, because a lot of logistic problems were there, bringing from each center. So what we have done from the regional compositing center itself, we identify 6 different parts, and then from 6 parts we have procured the meat with the different cuts of the meat, and the inland fish and shellfish also we have collected. And different cuts like chops, legs, brain, internal part also, the brain, tongue, lung, heart, liver, tribes, spleen, kidney, and tubes. All these different parts also we have collected for individual analysis. Coming to the marine fish and shellfish, actually we have identified 6 major fishing fish landing, or the the fishing center, where it's starting in the western side is Mumbai, Goa, and Kochi, and then the eastern side is Visakhapatnam, Chennai, and Mandapam camp. We went to the directly to the harbor where the fishing major fishing centers were there. There we have collected these marine fish and shellfish, and all this fish and the meat, particularly the boned meat, we have removed all the bones of fish and animal foods, and then only the meat part only we have properly frozen, and that was shipped to Hyderabad for the analysis. So as soon as the sample reached Hyderabad, and we have checked the integrity of the sample, in case if any unforeseen, due to some other reasons, if the frozen was not done properly, or if unacceptable for the analysis, if some other reason and the sample integrity is not good, so immediately we have discarded the sample, and again we have entered that for this, the next set of, or the next round of sample collection. And we have labeled properly with the alphanumeric code, and then stored appropriately at minus 80 degrees Celsius, all these samples. Or in case of the dry samples, it was stored at 4 degrees Celsius. Yeah, so these are all a few pictures, like ways to pack and proper packaging techniques, transfer as soon as transfer to the laboratory. So the very important thing is the some of the vitamins, the heat sensitive vitamins like vitamin C and folic acids, it needs to be analyzed immediately. As soon as it's reached, then we have to clean the samples properly, and then we started analyzing these kind of sensitive nutrients. So we have established a very good facility to conduct more than 150 nutrient component in a single umbrella. So we have um, state of art laboratory facility. There is no doubt about that. And we used most of the analysis. We have used the official methods, the association of official analytical chemist, or association of an association of analytical cooperation. And wherever the official methods are not there, so we have adopted, it is well accepted or internationally reputed or the published method, and that also we have re-standardized and validated properly. And all the methods, invariably the official method or non-official methods, we have standardized and validated using IUPAC or the UOC guidelines. And so more than about 200 discrete food constituents, which include all proximates, water soluble, fat soluble vitamins, individual free sugars, oligosaccharides, minerals, so on, so on, like that, we have conducted in the development of Indian food composition tables, as you know very well that. All the methods adopted scrutinized for the robustness, analyzing for the components of the interest in the food, in different food matrix, because the acceptability or the method, standardizing the method for different food matrix also, it is very important. So almost more than 2.5 years, close to 3 years, we spent only for standardization of these methods. You know, And for the different matrices also, the same method, what will happen, and we have to check different food groups. The method which we used for the cereals and legumes also, it should be acceptable for the roots and tubers where the more water content will be there. So based on these kind of matrix, different matrix specific adjustability, everything it was tuned before starting the exact or the original analysis. So these are all the different methods which were be used. It also is recommended by FAO, because there are different methods have been used for each analysis. But out of all the methods, the best method also have been discussed very properly, and those kind of methods we have adopted. And these are all the adopted method and which we have used for our Indian food composition tables. And if you look at most of the official methods, coming to the vitamins also, most of the official method we have used. And the technique, if you see earlier, uh, one example I can tell you that the earlier the folic acid content, it was used by microbiological assay. Though the microbiological assay to give the total folic acid or the free folic acid as a gold standard method, but now the sensitivity of the method is it, it's a better sensitivity we can find out in the LC method, liquid chromatography, or HPLC method, where we can able to quantify up to the vitamin level. Suppose each vitamin, let's say folic acid, there will be a more than 5 vitamins will be the tetrahydrofolate, methyl tetrahydro, formyl tetrahydro, and folic acid, like this. So these vitamins also can be able to separate and quantify using the latest technique. Similarly, the vitamin E, earlier it was quantified as a total vitamin E, but now we are able to quantify all the vitamins individually. Alpha tocopherol, all alpha, beta, gamma, delta tocopherols, and respective trienols. All these 8 vitamins in the vitamin E, we can able to separate and quantify. So the another important thing in the separate and quantifying the individual vitamins, the total vitamin E, it's not like the activity of vitamin E, it will vary depending upon the profile of individual vitamins. So that's the reason, alpha, beta, gamma, delta may not be having the active vitamin E activity, the similar activity. So there is a conversion factor for calculating the total vitamin E equivalents. So based on the calculation, and we have used the factor for converting the individual vitamins of vitamin E. So like this, similar kind of approaches, the latest approaches, whatever it is, thoroughly studied by various group of food component or the nutritionist or the food composition analyst. So these kind of methods we have adopted. And like that, free sugars, starch, and all the methods whatever we have used, the latest method. And of course the phosphorus, this is very one of the important parameter to be discussed in the today's the presentation of the meeting. And we use the calorimetry method, and is one of the gold standard method by the Fisky Suparao method. And the heavy metals and trace elements, of course, this is the best method to estimate at a very sensitive level, up to a ppb level. It's inductively coupled plasma with the mass spectrometry using AOAC method, AOAC technique. Like that, organic acid, oxalates, fatty acids, gas chromatography we have used. Amino acid is a very one of the best method we are using. It's a very robust method, exclusively for amino acid analysis. We have the machine analyzer, automated amino acid analyzer, and the phytosterols, then polyphenol also, individual polyphenol also we have quantified using HPLC technique, the phytates, saponins, like that's go on. So then another important the criteria when we develop this kind of the reliable database, we have to maintain the quality control and quality assurance. That is very important. We are continuously participating in the international proficiency testing by the WAPAL. Since more than 15 years, we are participating continuously, and we are getting a good Z score also. Apart from these IP program, we have used the certified reference materials or the standard reference materials, which are very expensive material, to validate our method. Validate the method in order to get our confident on the data. So we have used all this kind of quality control, not only these kind of CRMs and SRMs, because it's, as I said, it's very expensive. We cannot use it for batch to batch. So eventually we also have incorporated some of the commonly available samples to develop our in-house quality control samples. So after developing the in-house quality control sample material, we have used this in-house QC in each and every batch to get the, the confident, or the to check the analytical value it's correct or is going or out of range. So like this kind of quality control, even now also we are using in our day-to-day analysis. And these are all the different number of foods in each food groups, so starting from cereals and grains, it goes to the animal foods like animal meat and marine fish, shellfish, marine molluscus, and then freshwater fish and shellfish, and finally oils and fats, edible oils and fats, where in the edible oils and fats we have given only the fatty acid content, because we consider that the more than 99% it's of only the fat. This is just to representations why we are going for the compositing of the sample at the respective regional center. As I said, the 6 regional center, we have pooled the sample which we collected from the different sampling collection center. So the first reason is like we cannot analyze individually, because analyzing individually, the first thing, it will take a lot of cost. Analyzing all 107 sample, it's very difficult because cost is very high for each analysis. And another thing, when we wanted to give a representative of Indian or the national level data, so we have to systematically we have to plan. So what we did, so we, when we analyze the regional center, now at the same time, I forgot to tell one more important thing. um, when we brought the pooled or representative sample from each regional center, that was analyzed individually. Okay, so when we pooled the sample which we collected in the east region, that was analyzed separately, and similarly the West India analyzed separately. Likewise, all the 6 regional samples was analyzed individually. Then the mean of all the 6 regions, or in case in somewhere we could not collect some sample from all the 6 regions, so in those samples, whatever the samples wherever we have collected in each region or a number of regions, that was pooled together as a mean. And that standard deviation also we have given for your understanding, so that was given as a national representative data. So why we have given like that instead of analyzing all 107 samples as an individual? If you look at here, this is the wheat for iron content, the whole wheat data. If you see, when we analyze the East India samples individually, you will get 4.27 with 0.75 standard deviation. But if you look at the range, it is starting from 3 to more than 5, sometimes 5.46 also. The range is very huge. And similarly, in each region, the range of your value, it will vary. That's of course it will vary, because the sample to sample, the nature is differ. The ecological variation differ, and the biological variation will differ. So therefore, the variation will be huge. So that's the reason, we have pulled all together into the single sample, and in the each respective region, when we analyze there, you can see the range is very less, and your the error also will be reduced. Similarly, if you look at the zinc of the data, it's starting from the 2 to 4.6, and in sometimes the 5.6. But when we pull together, so you will get the range is very close by. No, so there are two important things. One is the cost effective, and another thing is the, in order to reduce this kind of error in the range or the variation of this kind of in order to reduce this variation, we have to pool it. So similarly, we have analyzed all the individual 6 regions for this kind of analysis: the proximates, water soluble, fat soluble, carotenoids. So I have given the table numbers and respective the nutrient profile. So as I think you might be understood from our Indian food composition tables. So this is a model page I just have given for your understanding. If you are not familiarized with this, and so the data which we have given with the food sample, food name along with the biological name, and the then along with this is a it's a in foods tag names. When we share the data with the other country, so when we give this kind of in foods tag names, it will be identified very easily. So it's one of the important criteria by the in foods or the FAO in foods. For example, here the protein or the fat is extracted, or this is a fat, and the first two three digits is belongs to the nutrient, and then the followed two letters, it belongs to the method which we have followed. So it's nothing but it's a fat, total fat, and it's a continuous extraction. What we have used, it's a continuous extraction, though there are three different fat extraction procedures, or the fat analysis procedures are there, like continuous extraction or mixed solvent extraction, and then the supercritical fluid carbon dioxide extraction. So we have adopted continuous extraction for the total fat. So like that, the inputs tag names also we have given for identification of the method with respect to the nutrients. Why? Because see, our I can proudly say that the IFCT have been used by many countries, not only India, many countries, and the FAO is recommending many other countries to follow our this kind of representation. And I also wanted to proudly say that this is the first food composition table in the world which contains as a completely analytical data. It's a completely analytical data with the systematically sampled and national level sampling. Now throughout the country we have collected this kind of systematic sampling, and the thoroughly or the complete 100% analytical data, it's other database doesn't have. Though the number of data or the food have been included in different databases huge, but the part of the data have been derived, as I said in the beginning. There are different ways of food composition tables we can develop. So imputed or the calculated, or even we can derived from the borrowed from other countries. So like that, so we have given our data in order to understand very clearly internationally or national level as well. And we also have given, sorry, yeah, here I think most of our users, they are having the problem to understand the data, because there are a plus or minus value also is there. So the plus or minus value is nothing but, as I said, the standard deviation of all 6 regions. So once again, we wanted to recollect that we have analyzed the data individually in all 6 regions individually. The south, north, east, west, center, and northeast. We have done analysis individually. Then we have taken the mean of all the 6 regions and the standard deviation of 6 regions. We have given as a national representative data. In case if anyone's required for in case of any specific foods for specific applications for the research purpose, any regional data is required, we are happy to deliver. So that also I just wanted to bring it to your notice, that is, if any specific foods, if you are looking for any specific nutrients, yes, we will be happy to share, if you wanted to for any R&D work. So that kind of, this is the representation of the mean plus or minus SD. And this is, as I said, in foods tag name, and this is to just to identify the page table identifier. Yeah, so we also have given all the names, not different language names, whatever it is there in our Indian rupees. Basically, we have given importance for those kind of languages. But I also tried to collect the script in each languages, but unfortunately, I could not collect. I collected the most of the languages, but I unable to validate it. But that the validated language scripts I will be including in the our databases. So now we are developing Indian food composition database where for the user friendly database. So that will be included the script of each languages as well. And that was released in the year of 2017, and that can be downloaded freely from this website. See, after developing this database, it was used by many applicants, many applications by different sectors like economists, agriculture planner, nutritionists, dietitians, the food serve managers, food agriculture scientists. Yes, now we are closely working with the food and agriculture scientists based on this database, and then food manufacturers, food technologists, teachers, and then home economists, epidemiologists, physicians, dentists, public health scientists, and non-specialist consumers, journalists, everybody they are following the composition, food composition table. It's well-known fact that. But apart from these kind of the directly usage of the database, based on the data, lot of using the latest technology like mobile application and web page, lot of modified versions of are they have used the database for so many other applications, like we can relate it to so many or the dietary intake or healthy apps or similar kind of apps. There are so many applications are coming, some people they have taken prior permission because the data is with copyright. So they have taken the permission to use the data, and many people they have not taken the copyright permission. But now we are we are identifying for those people to close their application. If anybody wanted to use applications, of course, they can write to us to get the right written permission, and then you can use it. But one thing is it should go as a free of cost, without any commercial background. That is one important thing. So we also have developed a mobile application because the book weight is almost more than 1.8 kilogram. So we wanted to reduce the burden. So we have developed our own mobile application. I don't know many of you are aware of not, and that is called Nutrify India Now mobile application, and that is available in both the platform, both Apple iOS and um, even the Google Play Store also is available. And we have included so many recipes also, and we can adjust the recipes with the different ingredients and the composition. So there are many applications are there, you can go through the. And also based on the data, we also have revised our recommended dietary allowances, which also published regularly by our NIN. And in 2020 we have released our revised RDA along with estimated average requirement. The first time in India we have included the EAR also for the public. And this is the template of the the web page. Already it is done, but the validation is under progress, and it will be released soon. And where more user friendly applications will be there, you just scan it, then you can understand the nudity profile, and different way of searching, by alphabetical search or the food group, the content wise, the QR code wise, or even if you are getting in the the bar diagram wise, whether in the ascending order, the descending order. So different kind of searches we are providing, and still we are working on this, because we are also taking if anybody having some different views or different idea, even from the public, we are getting some intellectuals Some people, they wanted to give their input, or they have some idea, some good idea which they wanted to incorporate it, so we are welcoming for that. So we are also still we are working on that to incorporate new idea that will be released soon. Yeah. So these are all about, the how we have developed our database and application of database, and then, based on the applications, what are the, subsequent development have taken place based on the, the latest technologies. All those things we have seen. But eventually, so there are limitations also there in the food composition database. It is internationally accepted. It internationally accepted those limitations are, first thing is, we have to understand that these food matrices, it's particularly our Indian food composition tables we have done on um, the fresh food. It's not a processed food, it is in the fresh raw foods. And these kind of foods, both the plant as well as the animal foods, of course these are all the biological materials. All the biological materials will exist some kind of variation, so due to so many reasons, the growing conditions or the soil types or the variety. So there are so many, the factors are there to give these kind of biological variations. In order to reduce these kind of biological variations only, we have pooled the samples with the respective regional centers, in order to reduce these kind of biological variations. That is one of the first and foremost limitation. And another thing is, um, the food composition table can be used as a device as a diet or meal supplement at the level, to estimate the nutrients. And another thing is that the metabolic studies, if you wanted to conduct the metabolic studies or the direct analysis of any particular food, and uh, better to identify, understand the exact nutritional compound of that food, it is advised, instead of going for this, the representative food data or the country level data, and better we have to go for individual analysis if you are planning for metabolic studies or any direct analysis of any particular food or nutrient. And the food composition tables cannot predict accurately the nutrient level in the foods. For example, the specifically for liable for vitamin C and folic acid, because you see sometimes, that's the reason I said in the food, as soon as the material reached NIN, we have started the analysis of vitamin C and folic acid. Sometimes what will happen, so these are all the foods which we collected from different sample collection centers. Then we have pooled, then followed by we have shifted. But if you look at the vitamin C content, sometimes if you are analyzing freshly collected food and these composite samples, sometimes yes, there will be a variation in terms of these kind of sensitive nutrients. So we cannot compare exactly, and that biological variations also is accepted. Then composition of given food may change with the, the manufacturer. This is for the processed foods. So my final slides, I can conclude that the current national sampling plan addresses the key foods approach identified in the Indian food composition tables, and but does not include the food specific to the subpopulation, because we are not concentrating on specific foods based on the subpopulation. And of course, this Indian National Food Composition Table program was initiated in 2011 with the objective of generating and improving the quality and quantity of the food composition in our country. And we have taken adequate infrastructure, we have developed in our NIN for conducting all this analysis. I also happily to say that our laboratory is a referral laboratory for international organization like FAO and WHO. We are conducting a lot of analytical program for them, and identification of key foods and statistically derived sample planning and analytical methods what we have used, everything it is, it's ideal planning for developing Indian food composition table. And we have analyzed more than 150 analytical parameters in each foods, with individually six representing regional centers, and then collectively we have given as a of our our technical team. And I would like to thank this team for their contribution and the support what they have done day and night, and weekly 6 days we used to work to continuously. And thank you so much.